Good morning, everyone. Welcome to Grand Rounds. Next week, our Grand Rounds speaker will be one of our own Columbia faculty member from the Department of Neuroscience, Rui Costa, who will be discussing learning new actions and shifting to automatic. I want to make one announcement that the American Psychiatric Association elections were held, as they are every year, and we had Phil Muskin was a candidate for secretary. He was in a four-way race with 3 men and 1 woman, and he won. So that must mean something about Phil's notoriety. So congratulations to him. It's a pleasure to have our Grand Round speaker come up from the Maryland area to be with us today. That's Dr. Antonello Bonci. Is it Bonci or Bonci? Bonci. Bonci. My Italian is impeccable. He's the scientific director at NIDA and also the director of the Intramural Research Program there. And his career has sort of taken an interesting path to get there. He's a graduate of Catholic University, but that's not the Catholic University in Washington, it's the Catholic University in Rome, the real Catholic University. And he received his MD there as well. He then did a residency in neurology and came to the United States to the Valum Institute at Oregon for a postdoctoral fellowship, and then returned to Italy for a 2-year stint before returning to the United States to work as a visiting professor in Rob Malenka's lab when Rob was still at UCSF, and remained at UCSF until 2010 when he, or 2010 when he was recruited from UCSF to NIDA. And while at UCSF, in addition to having worked with Rob initially, he gravitated from what his initial work was in the cellular pharmacology and pathophysiology of neurologic conditions to the cell and molecular biology of substance abuse. And that has evolved as the technology has become more sophisticated, including with the use of optogenetics, but also gravitated to not just beyond the effects of repeated administration of recreational drugs, but also the cognitive and emotional processes which drive addiction. So, Dr. Bonci is also a member of the National Academy of Medicine. He was elected in 2016. And I see he also got the Distinguished Scientist Award from my alumni, my alma mater, or so to speak, University of North Carolina, Chapel Hill last year. So congratulations. Please join me in welcoming Dr. Antonello Bonci.
Thank you for that wonderful introduction. It's a real pleasure, guys, to be here. I know that this is Grand Round Psychiatry, so most of my talk is pretty unusual in a sense that is much more focused on translational work that my lab has done, and I'm just going to have the first part, like the hardcore physiology, short and sweet without putting you to sleep, and then we'll move into a very big question on how we can take advantage of existing technologies in basic science and in the clinic and make sure that we as physician scientists or basic scientists take most advantage of what is going on right now as the scientific landscape has changed so much and so much for the better. So first thing I gotta say is that Jonathan has been a wonderful host, and I also scored big points with my daughter because she's pretty obsessed about Alexander Hamilton's play now. So yesterday I had to go to the store at the theater to buy a bunch of stuff for her, and now she loved me forever, or she'll pretend that at least. But I made her day, so it was really worth the trip no matter what. So I am, as Jeff was saying, I'm a very strange creature. I am a neurologist who loves psychiatry. I wish that psychiatry was what it is now 20 years ago when I had to choose a residency, because I am much more attracted to complex behaviors than to, let's say, motor neuron diseases. That's why over time I really shifted more toward substance abuse and complex behaviors as a scientist and as a clinician. And this is just a very simple picture of what dopamine does and what it's involved in. As you can see, it's kind of the perfect molecule to stay between neurology and psychiatry, right, with all these different and very important conditions and diseases. And so the main bulk of my work over the years have been done by using patch clamp electrophysiology. So I am, by training, a synaptic physiologist. This is a beautiful picture of my second wife, a dopamine neuron, which I married 25 years ago. I still love her. I still have no clue what it does, like in real life with real wives, but that's another story. So that's the primary technique, as I was saying, patch clamp synaptic physiology. We had two beautiful papers that are basic physiology, and I'm not going to talk about them now, just for the ones of you that are curious. One question was, still pending in 2017, what do GABA A and GABA B receptors do to modulate dopaminergic cells activity? GABA B receptors remind you of baclofen, which is a commonly used medications for spasticity, for example. Well, Nick solved that mystery. Don't want to say anything more, but just please read this beautiful paper that was just published a couple of weeks ago. Another big mystery that no one, I believe, had put together quite as well as Hugo is the many different roles and effects of kappa receptors. Kappa receptors are actually very important by causing dysphoria. They are relevant to many forms of substance abuse, as you know, to stress and PTSD. Well, Hugo has put together this paper, also published a couple of weeks ago, putting together a unitary framework in terms of physiology of kappa receptors into the nucleus accumbens. How do they modulate pre- and post-synaptic sites, and explain what happens overall in the nucleus accumbens when you activate these kappa receptors. So if any one of you is curious, please check this article as well. And my long-time work line has been to work on synaptic plasticity. When I was with Rob, we showed many years ago that dopamine cells, like many other places in the brain, as you know, the hippocampus first, do show long-term potentiation, right? This form of long-term plasticity produced on AMPA receptors. And that was no news, no big news. But my first postdoc, Mark Angles, showed that even a single cocaine exposure by itself produces a very long-lasting form of potentiation of these AMPA receptors. Single injection, cocaine is on board for a couple of hours. 5 days later, you still have this lasting potentiation of AMPA receptors. Then Billy Chen, another postdoc in my lab, a few years later, after 4 years, showed that when you don't inject cocaine, but instead you give repeated access to cocaine self-administration, now the length and duration of synaptic plasticity on dopamine cells, this form of long-term potentiation, is lasting for up to 3 months after the last cocaine exposure. This paper was conceptually very interesting because it tells you that it doesn't just matter about the pharmacology of cocaine per se, but associative learning is very important to maintain and strengthen these synapses for a very long time. If you think about it, 3 months of abstinence from cocaine in a rat is kind of equivalent to a few years in a human, right? So this is a very long-lasting effect. And at the opposite end of the spectrum, Garrett showed that when instead dopaminergic cells do what they're physiologically meant to do, to code for salience, you can actually see that LTP happens in the absence of any drug, just by pure learning associations. Only the day these were trainings for just a few days to associate a cue with a reward. And rats form LTP, produce this potentiation only on day 3 of this very simple behavioral protocol, cue and reward associations, when they actually learn something new. They learn to associate rewards with cues. But LTP, under physiological conditions, is only present on day 3 when they learn something new. Then physiologically, they reset themselves down back to baseline, which is kind of similar to what Rick Huganer has just shown during sleep, right? When you sleep, in this beautiful science paper, your synapses go back to pruning themselves and reset themselves to baseline. So happens in dopaminergic cells for plasticity you learn something new, done, then you reset the system back to baseline, ready to learn new information in the subsequent days and so on. But now, the big question was, okay, how come that physiological you have a one-day LTP, and with cocaine you have this very persistent, chronic, rigid, long-term potentiation? What is different between these two? So if we could turn back time, let's say we are all in the 80s, beautiful fashion and hairstyles, well, it would have been so much easier, right, to figure out the mechanisms that are underlying, for example, cocaine-dependent or learning-dependent LTP in these dopaminergic cells. But life goes on, proteins accumulate more, and this is a very kind of obsolete slide. So where do you start to try and figure out what does this LTP means and how can you study that in the context of cocaine in general and try to demonstrate what that is? So one target out of these many important targets that we chose was this ATPase called Thorase. This is an ATPase that Ted and Valina Dawson, about 6 years ago, brought forward at Hopkins. And what is interesting about this ATPase is that Thorase is responsible for removing subunits of AMPA receptors, the non-calcium permeable AMPA receptor subunits, from the membrane back into the intracellular space, okay? So it's responsible for recycling AMPA receptors in and out from your membranes. Yes?
Before you get into the time test, one question of clarification.
Yes.
The sensitization with repeated administration is basically what characterizes sensitization.
Yes.
So if you intermittently expose and then use weight and expose, there's an augmented and persistent elevated response. Is the cognitive acquisition also persistent, or are you describing both of those as being similar?
So the sensitization, if you do cocaine sensitization, as a matter of fact, Jeff, you get the same short-lasting LTP as a single injection. Even if you do 5, 6, 7, 10 injections of cocaine and you do see sensitization, there is a very tight correlation between locomotor activity and sensitization during the acquisition, but then they last as short as a single injection, 7 days. So you can keep on repeating to inject cocaine. When you stop, you have this kind of leftover plasticity for a few days, but it is not as learning association as cocaine self-administration, which is 3 months. So there is a very big difference between the length of sensitization-related LTP, a few days, versus associative learning self-administration LTP. Very different. So what we did with Thorase was to take advantage of a Thorase knockout and remove it from dopaminergic cells. And this is what Marco did by crossing the Thorase knockout with a DAT-CREAT, the dopamine transporter, okay? And by crossing them, you basically selectively delete Thorase from dopaminergic cells. The point being, now we are creating a situation similar to what we see after cocaine. You have this synaptic rigidity, AMPA receptors stuck on a membrane as a consequence of lacking this protein. And I gotta say, for many of you that have never heard about this, I bet 99% of you, this ATPase is a very interesting character. For example, there have been recent studies, Ted and Valina Dawson are following up on that there are families suffering from schizophrenia who also have mutations of ATPase. But we really don't know pretty much anything about this protein. So it's a very unknown target for all of us, right? Typical ATPase, short, A and B linker domains, but that's it. So first question that is fundamental for us as physiologists, what happens if you freeze these AMPA receptors on a membrane to their ability to signal? Are they strong? Are they weak? Are they non-functioning anymore? Or are they still active as if they were trafficking normally? And then what are the behavioral consequences if you have a model such as that? And the first control that Marco had to do was to test whether probability of release toward dopaminergic cells was changed. And in a nutshell, we can use a lot of electrophysiological tricks. This is one of them, to do per pulse stimulations. The point is that no difference in terms of release, no compensatory effects toward dopaminergic cells in terms of glutamate release. No effect there, which is good. But if you move to the postsynaptic site, to dopaminergic cells, then what you see is that AMPA receptors, this is one of the many measures that we collected, are actually in this long-term potentiated state. They are more active compared to control. So yes, these AMPA receptors are frozen into the membrane of dopamine cells, but they are perfectly functional, and actually they are producing this spontaneous at baseline LTP state. And nothing happens to the counterpart, the inhibitory receptors, which primarily are the GABA-A receptors. They are completely unaffected by Thores. This is telling you that Thores has a very specific effect. And by the way, we checked also on MDA receptors. On AMPA receptors, nothing on GABA and nothing on MDA. So it's a beautiful model to study behaviorally what happens to these dopamine-related behaviors in the presence of this LTP. And it's also very important to test what happens to our electrically induced plasticity, right? Because if you give trains of electrical stimulations to depotentiate these synapses, well, what you see is that they are not able to depotentiate. You cannot induce the typical long-term depression that we normally can in dopaminergic cells. This is telling you that these AMPA receptors are hyperactive, but they are very rigid. They are not flexible at all to move up or down. And the same happens to LTP. When you try to induce electrically long-term potentiation, these cells are once again rigidly into this up state, not able to go down in terms of activity or to go up in terms of potentiation. And that's the summary. So it's a very beautiful, pure model of AMPA receptors LTP. So what happens to classical behaviors that we can test on these cells? One of them is very basic locomotor activity in the open field. No changes there, not some generic random compensatory effects there. Another example is the elevated plus MACE test, another very general measure for anxiety. And once again, nothing really changes there. So what Marco thought was that, okay, we're talking about learning, we are talking about dopaminergic cells. Now let's try to do something that is involving a learning association once again. And the trial, the behavioral test that he chose was contextual and cued fear conditioning. This is a beautiful behavioral model because it's lasting like 5 minutes. And I know nothing about behavior, so I love short and simple stuff. So this is a very simple test, as a matter of fact. You put a mouse into this cage, right, where there is a tone. So he learns very quickly about the environment, context, and he learns about cues, which is a tone in this case. And during that learning phase, baseline, he will get food shocks. And that's the food shock. And then you measure the amount of freezing behavior following up that association. Cue, environment, food shock, they're going to freeze when they learn that this may happen again. And then you dissociate the two. The second day, you keep the same context, but you remove that cue, and you see how much context retrieves that memory, right? That's the test. The third day, you change the environment, you change completely the context, and you just keep the cue. And then you measure again how much they freeze. And to our surprise, they really learn this, and they learn it very, very well. They remember both context and cue very well, actually. They freeze much more than under control conditions. So these guys are, in a sense, much more sensitive to any form of cue, fear, associative learning, okay? Not only that, but if you follow the green dots, they learn it better, quicker, and they keep that memory up to day 11. So a very quick conditioning, but they maintain this fear, this association for a very long time, much longer than the other guys over here. So the reason why we like this is that this is kind of a very easy and simple way to test what plasticity does to one of the ways to test for PTSD or fear-related associations, right? Because clearly, this is making them much more, these cues much more memorable than in the control line. So what now the reviewers were asking us, which is a very good question, it took just a few months to do it, fine, that's a nice correlation, but show us that this actually means something, that you can rescue this behavior by reinserting Thores into these dopaminergic cells, and then that should make these guys less sensitive once again to these cues. So a few months later, poor Marco did all those experiments, and he basically injected the virus into the VTA, the midbrain, to rescue and re-express Thores into these dopaminergic cells. And first of all, as you can see, that increased activity of AMPA receptors is gone, so you are rescuing the synapses back to baseline from this LTP, and these are different ways to measure that. But also, you are rescuing this behavior, because during the training phase, now that you have your Thores guys back, the Thores is back into dopaminergic cells, they learn freezing as normal, but the amount of freezing now is significantly reduced. You reinsert Thores, they don't fear Q reward association anymore, Q freezing association actually. And the same is here. So that conditioning for the food shock is removed just by reinserting Thores, okay? So the overall idea of this study is that when you remove Thores, you produce both a selective potentiation of these very plastic AMPA receptors and a heightened ability of associate fear-related cues with dopaminergic cells. And when you reinsert them, you remove that kind of anxiety, PTSD-like behavior, and you can rescue that very selectively. So, but what is really new here is that we traditionally, as you know, think of dopaminergic cells in terms of reward, in terms of motivation, right? In terms of positive behaviors, but this is an aversive behavior. So it's kind of the opposite of reward, but it has very much to do with the other theory that we have, and many people have sustained for many years, that dopaminergic cells and dopamine is not really about reward or motivation per se, it's really about salience. What matters to you for survival, well, you gotta learn it quickly, and it can be either positive or negative, but it's very relevant for you. So removing just the reward side of dopamine into the salience role of dopamine. And yes? Correct. It is back to the wild type levels. Yes. Let me see. Here, right? Thores rescue, knockout and rescue, knockout and rescue. So they're pretty much going down. Yes, sorry. I'm going very fast because I want to get to the translational part. So just remember dopamine, salience, plasticity important, not about just reward and not just about motivation, okay? That is the main message. And now we are getting into translation. I'm so excited about this story. Diana Martinez, who's here, has been part of and helping me a lot to think about this in many different ways. But my lab has done in these last several years many, many studies related now to system neuroscience to really figure out in this entire limbic system how do single pathways in this VTA to accumbens, prefrontal cortex to accumbens, and VTA, all these pathways contribute to shaping cocaine-dependent learning and drug abuse-related behaviors. And these are all beautiful basic science studies, etc. But one of these studies actually set up an entire new line of research that went straight to the clinic within 6 months of the publication of this study. And that's what I want to tell you about. So the study that Billy Chen did was based on data by Nora Volkow and by many other clinicians showing that chronic cocaine abusers have a hypoactivity and in general a very important rearrangement of the activity of the frontal regions as a consequence of cocaine exposure. This has been shown with imaging studies and in several other ways. And what Billy did was to try and use a rodent model to try and model the human persistence of cocaine seeking to the best of our capacity. So what Billy did was to use this self-administration paradigm that Pierre V. Piazza, Barry Everett, and many others developed more than 10 years ago, where after a couple of months of cocaine self-administration, you introduce a few sessions where you give a food shock again. So you introduce a negative consequence to taking cocaine, right? That is as far as you can go to teach rats that, you know what, you take cocaine, we're going to punish you somehow. And what this protocol did, as it has done in previous studies, is to select these rats into two groups. 70% of them, 7 out of 10, we call them the shock sensitive, they would give up cocaine. Even after a couple of months, they thought probably wasn't worth it being shocked, okay? So they just no cocaine self-administration. But the other group that Barry Everett calls the compulsive seekers, they would keep on self-administering cocaine despite these 4 sessions where they were receiving these food shocks. And then we used optogenetics. And Billy chose these brain regions. This is called the prelimbic cortex, which is a portion of the frontal cortex that is affected by cocaine. And the prelimbic cortex is beautifully positioned because it's connected with the entire limbic system, with all the key brain regions that we really care about. And what Billy did was he injected channel rhodopsins into the prelimbic cortex to make sure that we could stimulate and activate these brain regions or inhibit them at will after cocaine exposure. But first of all, what happens to this prelimbic region after this chronic cocaine exposure in the in the shock-sensitive guys and in the compulsive guys? These are action potentials evoked from one of these pyramidal neurons in the prelimbic. There is a significant reduction already in the shock-sensitive guys as a consequence of cocaine self-administration. But in the shock-resistant, the compulsive cocaine seekers, you have a really additionally significant reduction in activity. This is perfect for optogenetics because all these guys were injected before starting any behavioral testing with channel rhodopsins in the prelimbic, so that what we can do at will is to use a fiber optic and turn on these cells here to make them look like these guys here and see whether that would reduce their interest in cocaine. And that is the hypothesis. If that hypoactivity is causally linked with cocaine seeking, then if you turn on these cells, that seeking for cocaine should be gone. And what Billy did was to turn on this optogenetic simulation at low frequency, just enough to turn on these cells without causing any seizures or anything. And then he measured again what happened to cocaine self-administration. And sure enough, the latency to press, when you turn on your prelimbic regions, the latency goes way, way up. Time between lever presses goes way up, and the number of lever presses goes way down, okay? So at this point, we're talking about April 2013. And what happened was that, this paper went out in Nature. There was a small daily newspaper from my hometown in Italy, actually, that interviewed me about this thing. Whenever I go back to my hometown, they interview me just to know whatever is going on with me. And they asked me about this study. And they said, okay, what's important about this? And because I'm optimistic by nature, I said, well, you know what? We could translate this into humans because you could use TMS as a non-invasive technique, transcranial magnetic stimulation. And maybe this is a cheap, available, non-invasive technique. Maybe these patients could benefit from it. It so happens that the father of a doctor who was addicted to cocaine, he had been addicted for about 15 years, saw this tiny newspaper, and it so happened that this doctor, this cocaine addict, just tried to commit suicide. He hanged himself in his own house the day that his wife told him that they were expecting a daughter. So this man, this doctor, felt that he was a threat to his own family, and when his wife told him about his daughter, he hanged himself, okay? His wife happened to run into the house. He was hanging from the ceiling. It's very big beams, old homes in Italy. He took him down, and the father of this man said, I will buy you a TMS machine, try it on my son. We have nothing left to do. That is how, in 2013, my fellow clinicians in Italy began stimulation with TMS on cocaine patients, right? And they started from the most desperate cases, from people where they've tried everything you can imagine, therapy, CBT, you name it, every possible medication, nothing had worked. And one reason why this could work is that, as I mentioned you, optogenetics was done directly into the cell bodies of these brain regions. So we didn't stimulate a single pathway. We stimulated the entire Christmas tree that starts from this light here, so that TMS is also a very general stimulation, but maybe there was a chance. And I'm sure that all of you know what TMS is, so I'm not going to dwell into this. Again, non-invasive external brain stimulation. It can be done through a figure-8 coil or a helmet, depending on the brain, and it has been in the clinic for many, many years. As a matter of fact, scientists started experimenting with electricity in the 1700s, and all this very cool experimentation with stimulating brains randomly, almost at the beginning, led in 1985 in England, in Sheffield, to start and using TMS stimulation for the treatment of drug-resistant depression. Okay, so this is a technique that has been around for 32 years, basically now. And one problem is, okay, TMS stimulation, but where do you do it? Is there an anatomical equivalent of the prelimbic region in humans? The answer is no. I tried that argument. They told me, stop it. There is no equivalent. The cells are very different, granular, non-granular. So I said, fine, I'm going to find another trick. So the trick I found was functions. What the prelimbic area in rodents shares with the dorsolateral prefrontal cortex in humans are behaviors. Okay, so they're both. This is a legitimate argument. Nobody has thrown tomatoes or eggs at me. The prelimbic and the dorsolateral PFC, which is, by the way, the region that we usually stimulate for treatment-resistant depression, share functions: inhibitory control in rats and humans, behavioral flexibility, decision-making. So we said, okay, let's go for the dorsolateral PFC in humans. And this is also a very interesting study. Alvaro Pascal-Leone here at Harvard in Boston and Michael Fox have shown over and over that when you stimulate, no matter which is your favorite site for targeting the dorsolateral PFC, these are 7 examples. If you stimulate in the canonically known locations for finding the dorsolateral PFC, if you don't have a neuronavigator, what you obtain with TMS is a very broad network effect, okay? Yes, you stimulate the dorsolateral PFC. In reality, you turn on the entire frontal region, okay? And the temporal regions and the associative regions. So it changes depending on exactly which spot you choose to target the dorsolateral PFC, but this is, guys, a network effect. So I'm not claiming, this is a disclaimer, not for a second that whatever we observe clinically is due solely to the activation of the dorsolateral PFC. We are using the dorsolateral PFC as an entry door to activate a network. Okay, clear. And the study was done in Padova by my colleagues, psychiatrists Luigi Galimberti, Alberto Terraneo, and our beautiful team. And we recruited for this very preliminary pilot study females and males, but the majority are males. And very important, these are all treatment seekers. And the majority of people here may know that treatment seekers have a very important feature. They tend to have a much larger placebo effect. They want to feel better. They have a lot of enthusiasm. We also know about medical-related placebo effects. Howard Fields and many others are highlighting this. If you explain the patient very well what the outcome could be, that can influence actually the outcome in terms of a therapy. So very prone to placebo effects. But also, this is typical of Italy. They had a guardian or guardians, the entire family, which was a strength, followed these patients very closely. We had this 24/7 monitoring on what was going on with these patients. So if they deviated from good behavior, we would be notified immediately, like mom or dad or sisters or everybody. So we knew what was going on with these patients 24/7. And in this pilot study, right, we had to provide this is an open label study, so very simple, very primitive, but general medical management, and of course, urine drug test as an objective measure, craving scales for cocaine, medical history, blah, 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 symptom checklist. So we did what we could in a reasonable amount of time without scaring the patients away. And while alcohol use disorders were excluding patients, of course, we excluded them. Just to be safe, we used anti-abuse in the patients just to make sure that they wouldn't develop seizures while drinking and getting TMS. So we stayed on the cautious side. And as I mentioned, these are very serious cocaine abusers, right? 16 years of cocaine used in the TMS group, right? 4 times per week plus. We started from the desperate cases. And the stimulation protocol, we kind of made it up based on what was known in the literature a little bit, but there wasn't much. We used the Montreal Neurological Institute coordinates, and we stimulated the left dorsolateral prefrontal cortex. 13 minutes total of stimulation, once a day for the first week, 5 days, then twice a week for the remaining 4 weeks, to assess safety, first of all, and feasibility in this population. And most importantly, if we got a glimpse of a signal, then to run with what is necessary, double-blind, placebo-controlled clinical trials. So this was the outcome of this tiny study. Control group, 16 patients, they received medications, whatever they needed, no side effects, 81%, 13 patients completed treatment, and 19%, 3 patients. Let's not use percentages. 3 out of 16, urine drug test negative. That's not really much. But in the TMS arm, and no medications, 11 out of 16, 69%. Then when we recycled these 10 patients who didn't respond to lack of TMS into this other arm, once again, 7 out of 10, urine drug test negative. So we had a glimpse, there was a light, there was a signal that this actually may work. The follow-up was 90 days, but as a matter of fact, the follow-up is 3 years. I'll tell you, we still have 70% of these patients who keep on showing up at the clinic. How much they lie, Jeff, we have no idea. 70% keep on showing up, we call them every month. So they keep on telling us, sending postcards and saying, I relapsed, I didn't. So we still have the majority of them, and we have 220 patients now just in Padova, and we have about 40 more around Italy in some other sites. But again, we gotta trust them a little bit, and when they show up, we test their urines, but we don't know what they've done for the other 29 days, right? So that's the reality. So, but as you can see here, proportion of cocaine-free patients, as you can see, 70%, but these are again small numbers versus 19, and the craving scale for cocaine went significantly down. Now, this is a snapshot into only 67 of the 220 patients that 3 years later we have in Italy. So this is just telling you that if you go, this is time, right, from the second month after the TMS protocol, 12 months later, and we actually have data now for the 36 months, you are beginning to see a spectrum of patients. Some of them, they needed a little bit more TMS. Some, like patient 14, kept on coming back because this patient said, well, you know what? I'm craving cocaine again. I want to be buzzed again. So that is good because some patients come before they crave cocaine. Some other patients show up after they relapse because they're ashamed of it, because they don't want to admit it. We have a whole spectrum of patients. But what is good in general, guys, is that they have no problem to getting more TMS. In general, they don't report any side effects. Quite the opposite. The side effects in this population, in many patients, is that they do sleep much, much better than before. Of course, their depression scale goes down, their depressive symptoms in many patients goes down. Many of them were pathological gamblers. We had 20 of them who actually said, I'm not interested in gambling anymore. That's it. These were people spending from 300 euros to 1,000 euros per day on average in gambling, their own homes, high-level professionals, very wealthy people in Italy. As a side effect, they said, but by the way, I don't want to gamble anymore, any money. I don't care anymore. So we are seeing a mix of anecdotal reports. Once again, take this very carefully. These are just opinions of patients. We have no proof 100% that it's the truth, right? They tend to lie as well, maybe to please us, to make us happy. So, but this is just telling you that you have an entire spectrum. And honestly, I would be very happy if we had to give 15 minutes of stimulation every week or 2 or 3. That beats nothing as an alternative, right? Because we have no neurobiological therapies against cocaine abuse or beats relapse into cocaine. Now, a year later, people are picking up on this idea. There is a double-blind study in Mexico City. We have collaborators in Chicago, in Boston. We are setting up a double-blind placebo-controlled trial at NIDA in my institute. Another double-blind in Italy, in Rome. There is a clinic in London who has been using our methodology for more than a year. They tell me that it's working. So we have a clinic in Switzerland, one in Cagliari, another one in, where the heck is in Milan. So even if you step out from Padova, St. Anthony, and the magic of this beautiful clinic, in general, what our friends and collaborators tell us is that 6 to 7 out of 10 patients after TMS, they do stop craving for cocaine. So, again, it's anecdotal. We need those double-blind placebo-controlled trials. And what else have we done? We got to figure out why it doesn't work in some patients, right? I told you 65% of these patients are responding to TMS, but what Tommy Raji at Northwestern has done is to collect all the data, because we use a neuronavigator, an MRI, with our patients in Italy, and check for every patient by cross-referencing urine drug tests with exact location of stimulation, who responded and who didn't. And this is an example of 20 patients. This is a paper that is about to be submitted, where actually was the main focus of stimulation in these 20 patients, right? Not exactly in the same spot. This is the average and the maximum intensity if you average all those 20 patients by crossing references with urine drug tests. But this is the most important slide, which I find it fascinating. This is the locus of maximum success if you were targeting that spot right here of the dorsolateral, but that is the worst. So if you are off by just a few millimeters, that has the lowest correlation with negative urine drug test and with patients self-reporting reduction of craving. This is just telling you that it's very, very important to know where we are stimulating. It's extremely important to use a neuronavigator to run MRIs and know exactly patient by patient what is going on. Otherwise, you may miss completely and have an opposite effect, right? Or nothing. God, I don't remember the anatomy. Does anybody remember what are we? What is exactly this one? I don't remember, Jeff. I gotta check. I gotta ask Tommy. But he said it's literally 2 millimeters north. So I'll ask him. Very important and clear message. So as I mentioned, it takes a village. We are running these double-blind placebo-controlled trials now, both in Rome. We are starting actually, and at NIDA. We are also correlating that with imaging that Elion Stein and Hyung Hang is doing. And we'll know the answer in a sham-controlled double-blind, hopefully soon. But the bottom line of this is that I was so happy that finally a rodent study got translated into a TMS study, into a clinical trial. We didn't have to wait 15 years. Again, we don't even know yet what is the mechanism that is changing in our rats the activity of these prelimbic regions. We have no clue on what are the molecular changes on that. But, you know, in a sense, as clinicians, if this is working somehow, we can do reverse translation and eventually figure that out, but move on and try to help patients and just try to make sense of what TMS does. And it says that TMS is safe and feasible in these patients. They don't hate it. They hate if you stimulate the right dorsolateral PFC. They will never come back. We tried that, and this is consistent with some other groups. Colin Hallon, Mark George, and others told me that the right is not equivalent to the left. And actually, there are asymmetries in terms of cocaine craving and inhibitory control after cocaine between left and right. But in general, our patients don't go to the right dorsolateral PFC. They won't come back. Urine drug test was the primary outcome. I want to use hair. We want to use other long-term objective measures for these patients to be clean from cocaine. Several studies are being developed, as I mentioned, and of course, 99% of this has to be done. We are also using rodent models of TMS to try and figure out the mechanisms as well. We want to do that. But if you think about this, guys, this is cheap. There is no IP, cost-effective. You could buy TMS in any poor clinic in the other side of the planet, and almost for free, you could treat hundreds of people that suffer from addiction, right? So this is an extremely not a good idea for a pharma, but it's a great idea for anybody who wants to start treating people that maybe cannot afford any treatment with cocaine and just get it done by a machine. It's just a few tens of $1,000s and get on with it, right? Once we know, of course, blah, 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 more controls. But the last lesson that I learned about this is that okay, TMS is right here. Optogenetics, this beautiful technique, super precise. I love Carl Deiseroth to death. He helped me to take this there. Precision, no idea, right? But guys, what's the lesson here? That we don't need precision. Precision allows you to formulate hypotheses. But when you move to a patient, stop obsessing, this is just my opinion, about, okay, can I really stimulate just the subcluster of pyramidal neurons, or how can I stimulate that brain region and maybe I'm going to see exactly the same outcome? The lesson that TMS is giving us is saying, hey, I am anything but specific. We have no idea, even if Tommy is computing every possible pathway that is activated by this TMS probe and whatnot. We are just guessing. We have no idea what we're stimulating, but it's working. These patients are alive. They come back. I actually interviewed many of them year after year after year, and they all tell me the same story. I was a dead person. I had forgotten my children. I had no life. All I wanted to do is commit suicide. I am alive 3 years later because of you guys. So who cares if we still don't understand the mechanism? People are alive, and it's our job to figure out why. Not obsessing about what is in common with optogenetics and TMS. Let's just figure out TMS now. So with that, I end, and I need to thank all the people in my lab. I just mentioned Mark Angles and Garrett Stuber and Billy's study, of course. Marco Pignatelli is responsible for the Thore study, and I have many other studies that I couldn't tell you about today, but the Carl Deiseroth, of course, for all the optogenetic work, and Luigi Galimberti and Alberto Terraneo, and the rest of the team on TMS for the second part of the study, and NIDA and State of California for funding in the past. Thank you, guys.
Thank you for really a fascinating talk, and it's clear that you're really a true believer. I guess, well, first question, is NIDA funding any trials?
No.
Why?
Okay. It's a delicate question, Jeff. So I am the scientific
Give
director.
you a diplomatic way to get out of it. They haven't received any applications yet.
No.
Okay.
They have. I am in a very delicate spot because this started as my idea. As a scientific director, I am not allowed to impose any of my ideas on any of my faculty. So when Elliot Stein came to me, said, Anto, I want to work on this, I was delighted, but I can't go to Nora or anybody else and say, you got to work on this. I'm in a very delicate spot because of conflict of interest. So if I say anything, that could be perceived as I am imposing my ideas on somebody.
But I mean, but they have to go through a peer review committee first. The review committees aren't giving them a high priority?
They didn't give them a high priority. I have a collaborator like Tommy Raj who proposed this work.
So
very low priority.
either they don't believe it or they don't want
Correct.
a competing hypothesis.
Yes.
Well,
Yeah, it's very sad.
so Diana, why aren't we doing this? Give Diana the microphone.
Oh, sorry, Diana. Don't want to put you on the spot. I didn't want to.
Well, we were one of the people that submitted a grant to NIDA with TMS and cocaine abuse, and it did not get funded. Oh, we did submit a grant to NIDA for this a few times.
And it didn't get funded
It
yet.
did not get funded. So now we're
that's
switching
horrible.
to chronic pain and Department of Defense.
Do we have any pilot data?
We do have pilot data, yes.
And
The pilot data is interesting. We actually did 3 groups. We did sham, low frequency, and high frequency, and sham and low frequency. And we use cocaine self-administration, so we just have people come into the lab and take cocaine as opposed to clinical trial.
In this same location, Diana, I don't know that.
We're using the H coil,
But the ACC or the dorsolateral?
the ACC,
So a
medial
different location,
prefrontal
okay.
cortex H coil.
Because our patients hated the ACC.
But we had a problem actually with a high frequency. We had a lot of dropout because people
Yeah,
found it
yeah.
very aversive. But once
Same
we
for
changed
us.
the parameters, we were able to get people through it.
Okay.
And the high frequency group actually increased their cocaine self-administration after a week of TMS, but then it decreases after 3.
Okay.
So 3 weeks of 3 weeks of TMS, they decreased their cocaine use or their cocaine self-administration, but 1 week actually increased it and was aversive.
So the ACC is the anterior cingulate, which is another region that is very heavily implicated, of course, in cocaine abuse. But, you know, if I were you, Dan, I would stick to what works. This is easy. Patients come back. I would move to the dorsolateral. Even if I love conceptually more the ACC.
We do have that coil. We do have the dorsolateral prefrontal cortex
Yeah.
coil. But we haven't had a lot of luck with the study section, so
I know.
I'm not
Well,
sure.
Terrible.
that's a implementation or issue. But so I mean, in mental illness, we don't have a lot of conditions that are readily animal modeled, but addiction is one. And so you've characterized the cell and circuit biology, and you've shown that hypoactivity in this anterior cingulate dorsal or prefrontal area in the rodent is correlated with this. And if you stimulate it with optogenetics, you can basically extinguish the cocaine-seeking behavior. So you've got a validated therapeutic intervention in an animal model. Then the question is, how do you translate that to humans? And one issue is the anatomy, and the other issue is how do you stimulate the activity? So your RTMS, which can either suppress or stimulate, is to stimulate it in this case. The problem with RTMS, as my biomedical engineer colleagues say, is the penetration and the impedance of the skull. Is that right, Diana?
It is one of the issues, and that's one of the advantages that we have with the H coil is that we do penetrate deeper into the brain. The disadvantage of the H coil is that it's more diffuse, it's less precise than the figure 8 coil.
Yes.
I mean, I think it's shocking that there's not more of an intensive effort to pursue this. And even if you do have a conflict of interest, I'm surprised Nora hasn't jumped on this and issued an RFA for something.
They have finally, actually, NIH with Josh Gordon, that you well know, and the entire NIMH and NIDA are beginning now. They are recruiting a program officer just to focus portfolios on brain stimulation. So finally, Jeff, things are beginning to wake up. It could have been done sooner, but it's finally happening. Let me just say that.
Well, therapeutic interventions for psychiatry get burnt because a lot of things that are reputed to work don't work. VNS is an example of, for what it's worth, I happen to be aware of the fact that in press in the New England Journal is a large RTMS study in depression that was carried out in Brazil, which demonstrated non-inferiority to Effexor. So if it's getting into the New England Journal, there's, I think, probably a greater credibility that it's acquiring. Sylvia, did you have a question?
Wonderful to hear.
Hi.
Hi.
Nice to meet you. As a fellow Italian,
Hi.
I really wanted to congratulate you for your TMS study because it's very neat, and also recruiting that number of patients and retaining it for that long. I know it's really challenging.
Thank you.
So I have two questions I wanted to ask you. One is, if you mentioned several brain areas and also several diseases that can be targeted from a behavioral point of view from different standpoints. So I was wondering if you have an hypothesis about what is that it's actually changing, if it's impulsivity, attention, learning. And the second question is about anatomy. It's true that there is no rodent model of the prefrontal areas that we have in humans, but we have it in non-human primates. And you could do something similar, although you don't have the possibility to target cells as well as in rodents, but you can do certainly, optogenetic or dreads to stimulate those areas. So I was wondering if you ever thought about making an intermediate models with non-human primates.
No, both very good questions. So let me start with the first. These patients, again, let's just pretend that they are not lying, okay? Let's just say that there are common features of them. They increase focus, they sleep longer, they feel less depressed, they show empathy. For example, cases where, again, even women with young children went to work, went home, changed, went out to get cocaine, ignoring like 2 years old and 4 years old. After TMS, they turn into stay-home moms just because they wanted to. They wanted to spend time with the children to make up. So there is a spectrum of changes, which is incredible to me. That's why I'm really insisting on the network effect. It can't be that the prefrontal cortex is coding for sleep. It's coding for cognitive features and empathy and craving and whatnot. Craving is the first thing that they say. They also talk about themselves as before TMS and after TMS. Before TMS, they say cocaine was my only obsession, my only interest, my only love, the only thing I thought about. And now I see myself. And that person, I just don't recognize myself anymore as that person. They can stay with their friends that take cocaine, and they are really bothered by that. So it is not that cues are evoking anymore an instantaneous craving for cocaine. So they report that. They also say, anecdotally, that the memory of cocaine per se is gone. And they challenge themselves. They want to feel that pleasure just thinking about cocaine. And many patients say, we actually don't. We can think about, we don't dream about cocaine anymore. We don't think about it because it gives no pleasure, to a point that actually a patient got really upset with us. He sued my friends. He didn't work because he was so upset that he didn't feel anything with cocaine afterward that he said, you took away the pleasure of cocaine from me. So upset with you. Yeah. So we've seen a spectrum of people. But there is another problem. You leave a void in these people. You remove cocaine from their life, you leave a huge hole into the daily routine. So we have been talking with many psychiatrists from around Italy to try to really come up with psychological support on these patients that are left without cocaine. They have a lot of time in their hands. Some people go back to what their life was, children, a job, their passions. They say, this cloud has been removed from my brain. I now care to read again. But some other patients are completely lost. And this hole that we leave after TMS has negative consequences of them unless we help them. The second question, which I don't remember anymore. Tell me again the second question. I'm sorry, I got lost into the first one.
I was wondering if...
Oh, the intermediate model. The only reason for why we are not doing that is because of time, because of cost. It would be beautiful to do it, but we have humans, we understand nothing about them, but we see some effects. And then we have rodents. We just don't have time and money and energy, but it would be beautiful conceptually to do it.
And I know you said that you don't want precision to be the enemy of the effective, but Andres Lozano, the neurosurgeon in Toronto, has been very adventurous in his applications, including addiction. Mm-hmm. Do you know if he's done anything that corresponds to this work?
I don't know. It's a great question. I don't know him, actually. I know Jeff Daskalakis and Daniel Blumenberg, we are talking about the cognitive side of this TMS, but I don't know.
No, Andres Lozano is a neurosurgeon. He's been doing DBS.
Yes.
And whereas most people confine their studies to depression or OCD, he's used it from everything from addiction to eating disorders to dementia.
Interesting. Yeah. I know that Christian Luscher in Switzerland is pushing this OptoDBS idea, which is wonderful. I believe that after 2 years, they have recruited 3 patients.
Yeah, it's slightly invasive.
Because if you can think, I would go through TMS first. And then
Ralph, Jonathan? Jonathan, Ralph, and then, is that Alex?
That was beautiful. I'm trying, so it's connected to what you just said about the networks, but also about this. I'm trying to get my head around this 2 millimeter distance change. So how do you, I mean, on the one hand, you're saying that we don't need to be precise and we can do this in clinics in Africa. On the other hand, if you do it 2 millimeters off, you do the wrong thing. So what's the trick to, other than getting huge numbers of patients and doing that kind of mapping, is there a way to use a network response to actually figure out in each patient? What's your way forward in trying to figure that location issue out?
Okay, yes. Step number 1, get a navigator and do, if ideally you could, Jonathan, an MRI patient by patient so that you stay south of that black hole, basically, the blue spot. The second is going to be that even if you try on a patient, and they usually tend to come back, these patients tell us, you know what, this is not working. So if we were in Africa, really, with no MRI, nothing, I'm making an example. In South America, let's say in countries where there's not a lot of money or an MRI, we could eventually, when we are more patients, try really to stay on the safe side in terms of the location instead of targeting the center of the dorsolateral PFC. That could be a way. But Elliot Stein and Hyung Han are actually mapping before and after TMS with a network every single area that is related to craving and not. So they want to try to use this as a probabilistic indicator on what are the regions that are being activated before and after TMS. So eventually we'll have much more precision than now. But beside that, if we didn't have MRI, we always take a chance, basically. That's what Tom is telling us. Yeah.
Well, there's actually, I think, a little bit more of a problem with that, because fMRI, if you're using bold activity, or even if you're using PET, which Nora has demonstrated in terms of the hypometabolism, the spatial resolution is probably not sufficient. But a neurologist here, Scott Small, has developed a technique that's gotten much less attention, where he uses a very high resolution, a acquisition sequence that has a very high spatial resolution and provides functional readout. It's a pre and post gadolinium test, and he's automated the parcellation of the regions of interest. So that would be something that would be desirable if you could do your mapping ahead of time. You're looking for the hypoactivity, and then at least begin targeting that region.
I'll find him. I didn't know.
Yeah.
Scott Small.
Scott Small, okay.
Yeah. No, it's a fascinating bit of work, so I congratulate you on the dazzling benefits. And besides location, the other question I had, because there was a group in England, I've forgotten who ran the group there, doing this, found that TMS dosing is also important, and that dose can affect memory. Now, you mentioned that maybe they forget, and the memory loss here is an advantage. But I know in England they were concerned, akin to ECT, that I wonder how variable the dose of the magnetic stimulation is a variable.
So,
Dose of treatment, and then the frequencies.
so for frequencies, for now, we have adopted only 2 levels of stimulation, the high frequency, 15 hertz, and the theta
15
burst.
hertz, I see. Yeah.
Okay, theta burst. And then we ask patients, what do they prefer, right? Some patients say that for them, theta burst is much better than the high frequency stimulation at 15 hertz. All of a sudden, they say, you know what, that was it. That really changed me. Some other patients, again, in the dorsolateral PFC, tell us the opposite. So this is, again, it's only anecdotal, but these are the only 2 frequencies that we have used so far.
So the patient has a dose choice, is what you're saying.
Yes, but it's still, we have only like 20, 25 patients. We haven't played with any other parameters so far.
And memory in terms of amnesia for something important has not been an issue. It's just that they forget their interest in cocaine. So that's a good memory loss, I guess.
Correct. No amnesia, but now we have set up a completely different database. It's all automated. We have moved to a new location. It's all on tablets. And we have 90 minutes of data collection, just behavior from memory, because we didn't do that at the time. We didn't know what we were going to find. Now we are. So we are taking care of all the cognitive domains at baseline with all the new patients for 90 minutes before we start any TMS.
Very important.
And then we'll collect that information. It's a great question.
Good.
Yes.
Alex.
So,
Alex, then Eddie, and then David.
so I have a mechanism question, specifically related with the memories and improving and getting worse. So in the beginning, you showed us that cocaine, especially together with associative learning, provides this long-lasting memory and form of plasticity, right?
Yes.
And then you showed us that Thorase manipulations was sufficient to reproduce some of those effects, while nothing else was. Is the Thorase somehow related to the cocaine? Does it work through Thorase? Is there any evidence for that?
So thank you for asking me. That is actually the next step. We want to train rats and mice to self-administer cocaine in this case, to see whether the inability to develop from baseline to LTP during training of cocaine is interfering with cocaine. That's why we started. We connected freezing of AMPA receptors that looks like what cocaine does in those self-administration guys is similar to having these receptors frozen. So we want to remove the ability to produce LTP, as in Thorase knockouts, from the equation and see whether they can learn to self-administer cocaine and if plasticity is lasting or if it's there at all. I'm sorry that I didn't say that, but that's the step that we are working on right now. Thank you for asking. Yeah, he's been waiting for.
Thank you, Jeff. Very interesting results, and so just one question about the clinical study. Did all the patients receive disulfiram, and in both groups?
No,
okay.
just the control group.
Okay, so which is interesting because disulfiram is an inhibitor of dopamine beta hydroxylase.
Yes.
And so could that be the effect that you're picking up as a confound, or is this effect essentially mediated by some dopaminergic, modulation by the disulfiram?
So if disulfiram was having an effect, we would have seen it on the control arm as well, which we didn't, right?
Oh, the controls did get it too.
Absolutely.
Okay, that was the question.
Sorry, I apologize. I went way too fast. So the control arm did receive disulfiram 0. But disulfiram, you're absolutely right, it changes also levels of dopamine in general, and TMS affects dopamine release, as we know. It affects probably BDNF, as Mart Sarma and many others are teaching me. So it may change activity of microglial cells. So TMS is probably turning on a big melting pot of many other substances which we have no idea about. But the disulfiram question, which is a very legitimate one, then we would have seen something in the control arm, which we didn't. Even in terms of, maybe you can ask me this, in terms of depression, does the improvement in depression change the outcome of abstinence from cocaine? The answer was no. There was no difference between medication versus TMS in our two arms, and yet the outcome in cocaine craving was massively different. So that's another answer to a question you haven't asked me, but sorry. Other questions?
Eddie,
Hi, excellent talk.
Thank you.
You mentioned that you excluded alcohol use disorders from the study. I was wondering, was there still any difference in alcohol intake between the two groups before the study? And then after the study, did you stop the disulfiram, and did you see a change in their alcohol patterns?
So during the study, in Italy, these guys that we accepted in the study were drinking alcohol in kind of an Italian way, a glass or two per day. 10 patients, if I remember, out of 18 that went through the TMS arm said that they drank less, whatever that means. Let's say from a couple of glasses to one a day. Cigarette smoking, 66% of these patients were smoking. We haven't properly studied that before. We had no clear reports on whether smoking was changing at all with them. But no, we haven't followed up afterward with disulfiram. We just focused on pure, as much as we could, cocaine patients for the other 190 patients. So we don't know the answer to that. Great question.
Our drug-addicted patients are nothing if not experimenters. They drive us nuts because they're always changing and trying everything different.
Yes.
So as a totally non-precise way, I mean, since this is inexpensive and they're going to kill themselves with drugs anyway, why not just give everybody a coil and tell them that there is, if you can find it, there's a sweet spot. Now you go and find it, you'll know when you find it. Your wife will know. Everybody will know. So why not just give one to everybody and let them sort it out?
It's what
CMS
do
doesn't reimburse for the costs of these right now.
Well, it's cheap, I mean,
well, not really.
Isn't it? How much does it cost?
No. It's about $60,000 per machine. If you buy, we have a B80 from McVenture, the cool machine. That's.
But look what we spend on.
Are you thinking of the direct cranial stimulation? There's direct current stimulation. DCS that's the thing you can give to everybody.
Yeah.
Right, then maybe that even.
Well, it's probably you get what you pay for.
Well, but kids buy that on Amazon and they use it to improve their video game experience, as you guys know. So TDCS is sold for just a few bucks on Amazon or whenever, and kids who play video games, they claim that it increases their cognitive performance or game. So in a sense, if we wanted to go TDCS, which by the way, in Brazil, there are colleagues that are studying methamphetamine and crack and whatnot who have been just using TDCS as opposed to TMS, and they claim that they do see some good results as well. But then you really lose completely specificity. But it's not a bad idea.
Even if not everybody had one, maybe if you just had it at the local clinics, and anybody could go in whenever they wanted and just go and try it, and you don't have to do it every week. You said you only do it once a week or a month.
There needs to be.
Yes. I did it on myself. I tried,
Yeah, you did? What happened?
Yeah. So I wanted to do it for a week because I couldn't. Nothing. But I did it for a day, and unfortunately, I'm not addicted to anything. But it didn't bother me. I was sitting on this chair for just a few minutes and staying there. I tried it because I wanted to improve my sleep. Sometimes I wake up for an hour or two at night, but I didn't have enough time to do it. But all of those kids and my fellow clinicians in Padova, they've all done it on themselves with these machines, and they loved it. Some people started sleeping better than before, actually.
Take away the things they like watching football or
no.
Listen, I don't want to sound like a wet blanket, but
there is so much bogus stuff that's advertised and promoted, and unfortunately even embraced by many of our colleagues for must be commercial reasons. And I think that it's really important, particularly for psychiatry, to be very careful and rigorous about what we view as really work. I mean, placebo is great in any form, whether it's TDCS or taking a pill or anything else, but these things need to be subjective. And we see it now because there's a resurgence in the use of recreational substances for therapeutic purposes, ecstasy, microdosing with LSD, and that's going to kill us. It's going to take us back to Wilhelm Reich and the orgone box, so we have to be, I think, very, very rigorous in how we evaluate these and comment on them. Anyway, that's my sermon for this morning. Who is next? Oh, Dick.
Right here? Yeah, Harold Sackheim, the sort of expert on ECT who was formerly at this department, originally conceived of magnetic stimulation therapy as a replacement for ECT, where you could be more stereotactic and use less energy, but still bring about a seizure. So as the field has evolved, it's sort of magnetic stimulation therapy has involved non-convulsive stimulation. But this begs the question, are there any studies with ECT on severe cocaine addiction? It would be preeminently justifiable given the risk-benefit, and also preeminently doable. You just do real ECT versus sham ECT. So the question is, what's out there in terms of ECT for severe cocaine use disorder?
That is a great question. I don't know, Diana, do you know? There's no studies. There may be studies in... So I'm going to be in China in just a few weeks because they contacted me. They want to start like massive TMS programs. So I'm going to find out what they've done. In Russia, probably it has been done, but I don't know of anything published. And Diana, you confirmed that.
I haven't seen anything published with ECT, no.
Which is kind of a pity because you're right. It does work. Yeah, I agree.
Diana, you want to comment on TDCS?
All I can say about TDCS is if you review the literature, there are 300 positive studies and 2 negative ones for everything ranging from math problems to sleep to Alzheimer's to drug addiction to Parkinson's. So I think larger, more properly done studies.
Well, what kind of journals are they published in?
Not
the best ones.
No,
huh?
but not the worst either. But there's an excess of studies using TDCS without any real great controls on how to do it.
Mm-hmm. And what do our biomedical engineering colleagues think?
They think it doesn't get through the skin.
What?
They think the stimulation doesn't get through the skin, much less the skull.
Right.
Yeah, I saw that study. Yeah.
Right. Well, you stimulated a lot of discussion, and we have to get on this. Look, just to give you some encouragement, Barry Marshall, the guy who got the Nobel Prize for discovering the cause of peptic ulcer disease, nobody believed him either for a long time.
So we are on the right track.
Yeah. Anyway, thank you very much. That was great.